Sorry, as I was saying, Dr. Amee Manges is a molecular epidemiologist and associate professor at the School of Population and Public Health at the University of British Columbia and supervises a research laboratory at BC CDC. She received her PhD in epidemiology from the University of California at Berkeley. Her interests are in human microbiome research and the application of next-generation sequencing tools to infectious disease epidemiology. Her talk tonight is titled Investigations into the Microbiome and Chronic Childhood Undernutrition.
Boy, I feel short. Oh, and I can see myself. Uh-oh. Is everything okay? Oh. Yeah. Okay. Good evening, everybody. Thanks, Will, and the VanBug group for inviting me to give a talk today. So as he mentioned, I'm an epidemiologist, so I have a little bit of a different perspective on the use of some of these next generation tools, both DNA, RNA, and metabolomics, and how they relate to studying diseases outcomes in humans. So that's been primarily my focus. I have one announcement to make. So I couldn't really do this project without the help of Dr. Ethan Goch, who is not here tonight. He was supposed to come speak with me tonight, but he had a family emergency and is in Belize right now. And Thaddeus Edens, who's a bioinformatician in my laboratory. So I just want to acknowledge them and also apologize that Ethan can't be here. So I'm going to really be presenting on some work that we did together and became part of his PhD thesis. So what's motivated this analysis is growth faltering. So here's a schematic of the growth deficits that occur in children in Asia in green and in Africa in orange. And it just shows over months of age of life on the x-axis and standard deviations in height from a WHO reference. So, when you grow, you have growth standards. You can measure whether you're at the 5th percentile or the 95th percentile. Well, this just shows you that there's a large number of kids in these areas that are 2 standard deviations below the average for their age. So you see this rapid decline, and there's evidence that the decline actually starts to happen in utero. And it's quite prevalent. So, in the project I'm working on currently in Zimbabwe, about 50% of the kids fall into this category of being 2 standard deviations below their normal height. Poor nutrition contributes to nearly half of all the child deaths under age 5 and impairs the physical and mental development of those kids. So it isn't so much that they're short, but there are cognitive implications for this poor growth and very, very common. And the other issue is that it seems to be an intergenerational phenomenon. So if you have low birth weight, smallish for gestational age babies, then they continue to grow poorly in their early life. They perform less well in school, which is partly related to the cognitive effects. They are shorter in stature as adolescents, and then for the women, they generally, because of their short stature, will then have babies who are then stunted. So this kind of cyclical thing occurs. And we really want to interrupt that poor growth. So there are lots of risk factors for linear growth faltering. So a lot of social, economic, and educational factors that are important. Obviously, you know of kids who have repeated bouts of diarrhea that can influence whether they grow well. Poor diet, obviously, and there's a phenomenon called environmental enteric dysfunction, which has been variably named tropical enteropathy, tropical sprue, environmental enteropathy. So the latest title as of 2 years ago is EED, environmental enteric dysfunction. And this is a subclinical pathology of the gut. It seems to be ubiquitous in people in the developing world, where you have a blunted villi in the gut, permeable intestinal membrane, and general local and systemic inflammation that results. And since it's ubiquitous in these populations, there's probably some adaptive explanation for it, but nevertheless, we think it's probably responsible, at least in part, for explaining the poor growth in kids. So obviously, if you have this underlying pathology in the gut, it makes it very difficult to, if you're getting marginal amount of nutrients, it makes it very difficult for the infant to absorb and utilize all of that to an optimal level. So, but the underlying etiology of that poor growth is not understood. The thought is that it may be acting through environmental enteropathy. Now, you'd think if you feed kids well, right? Seems like a simple explanation. If you feed them adequately, they'll grow well. Well, if you were to scale up all of the best interventions that have to do with nutritional interventions that have to do with stunting, probably only reduce stunting by about 20%. So there's obviously contributions from other areas. The other likely explanation would be diarrhea. But even if you were to scale up interventions to try to reduce the incidence of diarrhea, again, you probably get a less than 10%, maybe around 2% improvement in growth as a result of improving water sanitation and hygiene in the communities. So not alone. So the question is whether you were, if you were ideally to be able to do both. There's a trial that's ongoing right now that I'm part of. It's called the SHINE trial in Zimbabwe, and that's what they're doing. They're doing a factorial trial of a comparator group, supplemental nutrition, improved water sanitation hygiene, and then the fourth arm of the group is combined. You need both nutrition and sanitation present to achieve the growth recovery in kids. And it kind of makes sense if you think, you know, about the transitions in society, and big changes that you see in growth, in stature, probably happened because there was both improved nutrition and better sanitation in history. So maybe that's an explanation for why this intractable problem has been so difficult. So we have a potential causal model for this. And of course, so I'm studying the microbiota, so of course I put that in the center of my chart, naturally. So we have the infant intestinal microbiota, and you have kind of two possible pathways that are contributing to poor growth. So the first one would be through environmental enteric dysfunction, and it would have primarily an immunologic effect where this phenomenon of EED is redirecting energy from the host away into the immunologic response and away from the child's ability to grow well. And the other one is through the inability for the gut to process nutrients and absorb nutrients and utilize them properly, and resulting in poor growth. So is there some evidence that the microbiota is responsible for that? So it's not just the microbiota, it's probably a constellation of factors where the microbiota is probably important. We have some evidence, although indirect, of a role for the microbiota. So looking at WASH, improved water sanitation and hygiene, so a meta-analysis of randomized trials in that domain saw an improvement in height for age z-scores. That's what the HAZ stands for, height for age z-scores. And then antibiotics, we did a systematic review and analyzed individual patient data from 5 randomized trials. And it looks like there's about a 0.04 centimeter per month improvement in kids' height when they're exposed to antimicrobials. Now, they were exposed to antimicrobials for many different reasons, but nevertheless, combining all of that information together suggests that there's an improvement in height. And there was an improvement in grams per month also, for almost 24 grams per month in weight gain. Then some studies of probiotics and prebiotics on the bottom here, looking at, again, systematic reviews and meta-analyses of randomized trials of probiotics and prebiotics showing an increase in weight gain in kids. So we were interested in trying to identify which microbiota bacterial genera might be important in linear growth. And so we did a secondary analysis of 2 very prominent recent publications, both from the Gordon lab. I don't know if people are familiar with Jeff Gordon's group, and they've done a lot of work on both obesity, so overnutrition, and undernutrition. So one trial of kids with kwashiorkor in Malawi, and also malnourished kids in Bangladesh. So we had access to 2 data sets, and this is important because when we first started doing the project, we started with just the data in this study, and this one was published subsequently, and so we were able to validate the observations that we're making. And if you're an epidemiologist, as I was talking to the students before this lecture, epidemiology has generally had I don't know, it doesn't always have the best reputation, because you have people you have news broadcasts 1 week suggesting association with one exposure, and then 2 weeks later you'll get contradictory reports. A lot of things have changed in the field of epidemiology, just for your information. So there's a lot of things that actually the basic sciences could benefit from some of the reforms that have made to the discipline, including the use of evidence from randomized trials, using systematic reviews, using all kinds of guidelines for the reporting of study results, having studies post their primary hypotheses, their primary outcomes in explicit detail, and all of their analysis plans in advance of doing the study, right? So no cherry-picking information after a study is conducted. So just little things that have, to try to sell my sell my discipline, that have improved. And some things actually could be portable to some other fields. So the published analyses of these data from the Malawi cohort, they looked at kids with severe acute malnutrition and its association with compositional differences in the gut microbiota as measured by diversity indices and functional gene abundances. And then the Bangladesh cohort, they looked at weight growth associated, again, with compositional changes in the microbiota. And both studies were actually found that the kids who were not growing well, who were malnourished, had immature microbiotas. It's as if the kids were, maybe they were 14 months old, and they had a 6-month-old's, nearly a 6-month-old's microbiota in terms of its profile and function. So they were suggesting that some of these kids who are experiencing malnutrition and are not going to grow well have an immature microbiota. So I'm stepping into a field where there's been a lot of amazing work in basic science using germ-free animal models. So most of these involve beginning with animals that are, devoid of any microbial exposure from birth. Then usually the comparison is that there'll be a conventionalization of that animal. So an entirely new microbiota will be, the animal will be exposed to an entirely new microbiota, or even a mono or bi association. So you may see studies where they look at a single microorganism or a couple of microorganisms and see what the effect of introducing just one or two on the animals. And through the context of all of these studies, and they've been done in fields in nutrition and immunology, et cetera, very vast array of studies, you find the microbiota is responsible for almost everything. It can be sometimes very confusing, but of course these factors themselves are interconnected, so it's not so surprising. So again, ablate intestinal barrier function or impacted innate and adaptive immune function, host metabolism, so there's been a lot of research on obesity primarily, but also by Jeff Gordon and some other groups on undernutrition and malnutrition, colonization resistance, so the ability of the gut to resist infection, even osteoblastogenesis, so this is the production of bone, or bone metabolism and bone mineral density, which is of course interesting to me, and even cellular stress responses. So as an epidemiologist, I'm coming into this field. I have a lot to work with. Unfortunately, the information and the inferences that are coming from the germ-free models, as powerful as they are, probably what we're looking at, it's more likely, is that you have a microbiota that is either depleted or enriched in certain organisms. There isn't an absence of a microbiota. So these kids, in my case, the kids that I'm studying have a fully functional, albeit maybe not optimally functional microbiota, and it's interacting, those microorganisms in that community are interacting with the host in a way that may not be optimal for growth. So we have this situation where it's sort of the opposite. What we really need to see is if you have a fully constituted microbiota, how can you alter that in certain ways so that it's not going to function in a way that it needs to for the child to grow and develop cognitively in its life. So I want to talk a little bit about the conceptualization of the microbiota, because there's a few different ways that people are dealing with this very complex community. Again, just to be clear, I'm sort of focusing primarily on the intestine. There's communities all over the body, as you know. So often you have a situation where the microbiota is sort of conceived of an organ, a unit, right? And so the individual characteristics of it are not as important. It's either dysbiotic or not dysbiotic, for example. And that interacts with the host, again, as an entity in and of itself and may be impacted by environmental factors. And we see a lot of analyses using principal coordinate analysis, clustering analysis, and the sort of characterization and definition of enterotypes, in my case for the intestine. But in reality, probably it works something more like this, where you have influences from the external environment, influences from the host, whether it's the genotype or metabolic processes within the host itself. Then there's probably interactions between the individual members of the microbial community itself. So much more complex, and I don't think I'm not necessarily sure we're doing the best job we can at trying to capture that. Now, of course, it's just another system in biology that is more complex than you really want to grapple with. But nevertheless, this is probably the reality, and this is the environment we often find ourselves in with the tools we have to do the analysis. So here's just an example. This is a study of HIV positive and negative individuals exposed or unexposed to antiretroviral therapy. And so you can see, this is just a typical report where you see richness in terms of the observed microbial species that were identified, and then a Shannon diversity index, for example. And then you'll see a principal coordinate plot where the colors here just indicate the groups. And it's just saying globally, looks like the microbiota is partly recapitulating the groups we already knew about to start with. So you can tell from the tone of my voice that these pictures sometimes are not very satisfying to me, because we knew you had people from long-term diet study, we knew we had people from the global gut study and the HIV study, and it's likely that their microbiota differ, but it's not telling you very much about how or why that might be the case. So in an effort to move away from sort of that organ perspective on the microbiota, we're going to try to use some other tools to help understand how they function. So the other big, the organ versus kind of modeling as a complex community issue is one thing to start with. The second big issue, and everybody in the room probably has dealt with this to some extent or not, is the curse of dimensionality. So again, I'm not generally working in an experimental setting, right? I have to study people. People are notoriously variable. They don't always do what you ask them to do. They vary in their exposures. They vary in their history of exposures in their whole lifetime. So add in all of the noise that comes from their collective life exposures, and then add in all of the factors that can vary in terms of the microbiota. So all of the, let's say if we just talk about bacteria, there may be in the gut 500 different species, right? How can you pull together a very noisy system with multiple variables and come up with a tractable statistical model in order to make some meaningful clinical inference? Right, so so metagenomic data and many other classes of biological data like this are high dimensional, where the number, in my case, of microbial covariates of interest are vast. And here in our typical toolbox for epidemiology, epidemiology is multiple regression usually, and the number of parameters being estimated far outnumbers the number of data points available to perform the estimation. So because of this situation, and again, other people are focused in this, I'm happy to talk more about it after the talk because thinking about ideas for how to handle this is very important. I need to reduce the dimensionality. So let's say just looking at the microbial taxa that are showing up in my study that may be important, how do I pick which ones are really influencing the biological process under investigation? So how can I, extract features? How can I choose the most important members of that community and then follow through with a more standard statistical model where I can take into account people's age, their underlying disease status, their exposure to specific drugs, et cetera. So all of that clinical epidemiologic information has to be modeled also in addition to the metagenomic or microbiome variables. So you have some options. Penalized regression, so the top two are examples of that. Ridge regression and lasso, and then random forest is another method which is based on classification. So all of these all of these are ways that you can try to figure out which of these variables, which of these organisms may be important. We propose to try a network analysis. So this has been used a lot in genetics and neuroscience. And we thought, well, this seems reasonable because here you have a community of microorganisms. Some of them are probably working together, probably not all of them. But if you presume that they are functioning possibly as a network, then that seems like a reasonable structure or framework to put in as a tool for your analysis. So we used one method. It's called lasso, which is a graphical form of lasso. And I'll talk about that a little bit. But first, let me talk about the networks. So people here, I'm sure, are familiar with the idea of networks. So this is, in my case, the non-random co-occurrence or co-occurrence of patterns representing some underlying relationships between those organisms in the microbiome community. And we can represent those relationships as a network. And it sort of reduces that complex system that we find very difficult to handle statistically into an abstract structure. Then you can operate on that structure and try to gain information that will tell you, okay, which members are important or which relationships are important. So the green, obviously, are the nodes representing the microbial taxon. The edges are those purported interactions or relationships. So one other element of networks is there are metrics that have been developed for years that try to characterize those networks. So for example, you have global metrics that try to capture information about the entire network. So that would be, for example, graph density. So I'd be looking at the probability that two randomly chosen nodes or taxa are connected, and measures the connectivity and the potential for information flow. So if you can have a really dense network or a very loose network. And then a local metric would be something like normalized degree centrality. So this is the proportion of other nodes a single node is connected to, and it might measure node importance. So for example, that would be the metric that might pull out specific microbial taxa that are important in the model that we're studying. So we tried to apply these metrics to the data sets that we talked about. So just to be clear, so you have a set of ecologic relationships that exists in the world, in the model that you're studying, and you're going to use the correlations between taxa that exist at the time points that were measured. So some are going to be highly correlated, some will be less correlated. You can take that correlation matrix, and you can produce a correlation network model that represents that sort of idea that you think is underlying your system. So we used an algorithm called lasso to estimate the inverse covariance matrix that captures that relationship structure using the data from the two studies that I mentioned. And elements of the inverse covariance matrix measure the magnitude of association between these taxa, adjusted, and this is important, it's adjusted for all the other variables that you think are important. So it's actually not just the taxa, but it's also the age of the child and the underlying nutritional status of that child, whether they received a therapeutic food or not, for example. And they weak or unimportant associations then through lasso are shrunk down to 0. That's the piece of the penalized regression. So that's the one that's limiting the number of variables or taxa that may be important. So the data sources, as I mentioned, we had the Malawi cohort and the Bangladesh cohort. So the Malawi cohort with metagenomic data included 22 twin pairs. And because the twins were followed up at multiple time points, we had 308 metagenomic data sets to work with. And Bangladesh, there were 11 twin pairs and 1 triplet. And so we had that data set. I think they were followed up sometimes up to 17 visits. And so we had 429 metagenomic data sets. So you'll notice, again, the unit of analysis here are the kids. So 44 kids, 25 from Bangladesh. So not very many individual kids, but lots of information from multiple time points that, for which the kids were the stool was sampled and sequenced. So we broke our analysis up into two parts. One was a nested case control analysis, and that was using networks to try to figure out which microbial taxa may be important. And the second piece was the longitudinal analysis. So this involved testing those selected variables from the first stage, the nested case controls, testing those selected variables for their influence on growth. So the longitudinal analysis included the use of a between and within twin regression model to adjust for confounding. That was implemented using generalized estimating equations. And we also lagged it. Since the kids had multiple visits, sometimes up to 17 visits, we had the values of whether the child had diarrhea, for example, or things like that, were in the visit before the growth was being measured, right? So you have the temporality was included. Now I have to say the original, and this is not a knock on the guys who did all the great mouse work, but in their analysis of the kids, they wanted to select twins, which totally makes sense, right? Twins are going to, in a sense, control for the genetic component, right? So you have twins who are discordant for malnutrition. Then you can try to see whether the malnourished kids and the more healthy twin pair, you can look for environmental factors that may be influencing why they are discordant. And that's great. But they didn't actually take into account that the twins are dependent, right? So they analyzed all the kids as if they were independent observations. And of course they're not, because the twins, the pairs, lived in the same household. So they're going to share a lot of the same exposures. Anyway, so I think this was a great, this is, as an epidemiologist, if I could go back to their group and say, I think you could redo that analysis using a different model that would take into account the between kid and within the individual kids and between the infants in the twin pair. I think that would be much stronger. Okay, and we also used false discovery rates because we were doing lots of comparisons. However, the sample was small, so we gave ourselves a little extra leeway by giving our alphas 0.1. So just a reminder, 22 pairs from Malawi, longitudinal samples. They did 454 sequencing on DNA that was recovered from the fecal samples. And this came up in our discussion again before the session. There's no ethically feasible way to get mucosal biopsy samples from kids anywhere, forget about in the developing world. So we have to use stool as a surrogate for measuring what's going on in the gut, and that is not ideal. I will say it right from the get-go, it is not ideal, but it's really the best we can do. And even more ideally, it would be great to be able to have a sample from the small intestine, but we don't. So there's always a little, the inferences that we make, we have to consider that fact. So height for age z-scores, weight for age z-scores. Again, this is the deviation from the WHO standards in height and weight in months and diarrhea in the 7 days. And they used metaflan to characterize the composition of the microbiota from those samples. And then in Bangladesh, again, longitudinal study, they used 16S sequencing. A lot of the same data was available. So just another note, the kids that were included in these studies not only were malnourished, they were really, really stunted. So this is just a picture of the height for age z-score distribution of the kids in Malawi and in Bangladesh. And you can see, I said before, kids are considered stunted when they're 2 standard deviations below their normal, the normal growth pattern at WHO. And here you see the vast majority of them are 3, 4, almost 6 standard deviations below. So these kids are very small. So for the purposes of this study, we actually had to define our cases as severely stunted versus not so severely stunted. So again, we're limited by the fact that we don't have a true pure control group. But that's just the reality that the kids are really suffering. There was a couple of other things that we had to do. So for the nested case control study, we had to select kids. We didn't want to, again, because the twin pairs, there's some dependency within the twin pair, we had to be careful when we were selecting cases. So we had to break apart the cases for the network analysis. So ultimately for Malawi, we had 10 cases and 10 controls. Again, this is just for the first step. So this is the case control network building piece to figure out which microbial taxa may be important. So we had 10 cases in Malawi and 8 in controls in Malawi, and 6 cases and 5 controls in Bangladesh. So we ran lasso and mapped the networks. So even though the numbers are small, we're collapsing over all of those kids, right? So the networks here are the combined networks and cases and controls, and you have the node size corresponds to the abundance of those organisms. So you see Bifidobacterium is expected to be a large group because the infants are generally, especially the younger infants in the studies, are being breastfed, and so that's the organism that consumes human milk oligosaccharides predominantly. And so you have the case and control networks, and you have on the left, you have the anaerobes on the right and the aerobes on the left. And you see the relationships that are that exist between the aerobes and the anaerobes. And you have down-regulated in or lower in the cases from controls with dashed lines, and solid lines were positively associated. So just another way of viewing the data, and using the metrics that I mentioned before. So, for example, how central the nodes are. We're going to use that information to try to extract from these networks which organisms we think may be important for stunting. So looking at cases, again, severely stunted infants, controls, not so severely stunted infants. We find, this is going to come up in a second, but Acidaminococcus here associated with cases or more severe stunting, because it was a highly central node within the networks. So just a note again about the within regression models. So again, we're interested in generic with significant differences in centrality or relative abundance. So that's how we decided of all of these taxa, which ones we're going to consider in our real statistical analysis of growth. And then we fitted a separate model for each of the genera by GEE, corrected for the false discovery rate. And this is just the model for the between within regression. So you have the term for the genus relative abundance in each infant, and here's the average mean genus relative abundance between siblings in a twin pair. So you have the within and the between terms in the model, plus a bunch of other covariates, including diarrhea and alpha diversity we adjusted for, and then the Z, the age and the follow-up time. So all of these variables, diarrhea, the alpha diversity, were lagged by 1 month, 1 month before the visit where the growth was analyzed. So here's just a schematic. So we have a twin pair, and if there was a difference in abundance of specific microbial taxa in twin A from twin B in the visit before the one we're using for our growth, and then we look at that next visit at whether there was a decrease in height for age between the two twins, we come up with Acidaminococcus. So Acidaminococcus was chosen to include in the analysis because it was a highly central node in the cases from that first stage of the analysis. We put it in the longitudinal analysis, the second stage, and we have an association with growth. Let me just read you the interpretation of this coefficient because it is so beautiful. A 0.01% difference in the relative abundance, again, these are relative abundances, so the numbers can be quite small because you have a lot of taxa composed of that 100%. So the 0.1% difference in the relative abundance of Acidaminococcus between co-twins was associated with a 0.08 lower height for age z-score in the next visit, at the subsequent visit, in the co-twin who had the greater Acidaminococcus, right? So they had the greater Acidaminococcus abundance compared to their sibling. And this was after adjusting for multiple hypothesis testing. So let me just read it again. So the 0.1% difference in the relative abundance of Acidaminococcus between co-twins was associated with a 0.08, that's this coefficient, lower height for age z-score at the subsequent visit in the co-twin who had the greater Acidaminococcus. So this is relating the presence of the microbial taxa and the subsequent growth deficit in one of the twins. Okay, what do we know about Acidaminococcus? Almost nothing. So looking back at the literature, trying to figure out, well, you know, after going through all of these stages, looking to see if there was some, you know, maybe something we missed. We looked at big differences in relative abundance. We looked at these network metrics to try to figure out which ones were important. We got Acidaminococcus and a couple of other ones. This was the one that, I should mention. So we did this for Malawi first, as I said before, and then the Bangladesh paper was published. We got the publicly available data from that study, redid the entire set of analyses, sure enough, saw the same relationship with a slightly bigger deficit in height for age z-scores. So it's just important to know. So this is 2 independent tests of it, which I think is probably a pretty good idea given the amount of noise and the possibility for false positives. I think it's great if you have other samples that you can test in. So the one thing that we found that was interesting about Acidaminococcus is that it was able to consume glutamate as its sole source of carbon and energy. And glutamate is really important for intestinal barrier function. And it's really important in the metabolism of other amino acids. So we think, okay, this is interesting. Maybe we're getting this signal from Acidaminococcus, maybe not because it in particular is so important, but it's representing a pathway. And because this is a sole carbon source, maybe we're seeing the signal because it's a sole carbon source. But there are other organisms that can do this. But we may see this one just because it can use that exclusively. So we formed this secondary hypothesis that glutamate fermentation may be one pathway, or it's related to other pathways that restrict growth in kids. And so we used this using KEGG. Since we had metagenomic data from the Malawi cohort, we went back and said, okay, let's look at the glutamate pathways. Let's see if we detect the genes from that pathway and see whether that differs, right? So we selected the first 2 genes, looked at the relative abundance of genes encoding those 2 genes in that pathway, and redid the analysis for just these 2 pathways, and so we see. Again, not a perfect recapitulation of this, but just testing, is it Acidaminococcus or is it the pathway underlying it that may be important? So again, we saw, excuse me, a decrease in height for age z-score associated with these 2 genes. So the summary, Acidaminococcus abundance associated with future linear growth deficits in already malnourished kids, and the biological markers for the potential for glutamate fermentation are associated with those growth deficits to be determined in some follow-up studies. But I think okay, so we have so just to acknowledge Ethan and Thad and our funders, the Vanier, Ethan had a Vanier Fellowship for his PhD, and the Bill and Melinda Gates Foundation is funding us now to do metagenomic sequencing on a sample of infants and mothers from that large trial that I mentioned before. And so I think the take-home message is there's a lot of work that's being done in experimental models, very important mechanistic studies that are being done. I think we need to kind of flip them and say, if you have an intact microbiota, can you selectively eliminate or enrich for certain members of that community and see what effect that has on intestinal barrier function, immune activation, things like that? Because the absence of a microbiota really changes things dramatically, and even reintroducing that later in the animal's life doesn't become an intact functioning organism again entirely. So flipping that around a little bit, and then asking from the epidemiologic side, right? You have to study people too. You can do the experimental investigations. You can do randomized trials in people, which I think are a real strength. So if you know you have interventions or treatments that you can do that you know will lead to changes, that's a great experimental design to do in people. You have, of course, some limits. You can't do anything to people, generally speaking. But being able to do that, make that perturbation and be able to measure the effects of it is really important as a way to validly connect microbiota function to some disease outcome. So I think that's the piece of it. And then for people who are doing just regular epidemiologic studies of the role of the microbiota in lots of different health outcomes. We need to think better about when we're sampling people, the context of the study design, and then also how we're going to collapse all of this very high dimensional data, because you do studies in people, you may be able to afford to study 400, 500 people. It's quite limiting. So given all the noise, given the wealth of data that's available from these new tools, you need to somehow condense it in a meaningful, rational way, and then also integrate it with other epidemiologic and clinical data. So I think that was something new that we did in this study, and maybe it will be useful if you guys are doing studies that involve clinical samples or epidemiologic cohorts. Anyway, yes, so thank you for listening. I'm happy to take questions. You have 6 questions?
Yeah, because I study.
Yeah. Well, I know you do.
Yeah, so maybe I'll let someone else ask.
Okay.
Too many.
You could take everybody else's slots. They'd probably be happy, unless there are other burning questions.
Sorry, it's very specific.
Okay. Yeah. 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 Okay, so the cartoon I put up was sort of just to explain where you can have metrics, or you can understand the network globally, right? How dense it is, for example, or sparse it is. And then you can have metrics that look at different, how central a node is, or how the length of the connection. So you can have nodes connected, you may have 5 nodes connected, or 2 nodes connected. So all of those things are things that they're metrics you can use to describe a network, and there's lots of them. So we had to choose a metric of the many of them that are available that we think would be important. So we thought, well, if the node is very central, in other words, like it's a hub and spoke kind of relationship, maybe we'll say those are important because they seem to be connected to other taxa. And just a point to make about that, those edges, many of those edges are spurious, right? They're not, we have to sort of apply this abstract idea of networks to try to organize the information that's being derived from the covariances of those taxa. But in fact, some of those edges are meaningless. But we're saying, okay, let's look at node connectedness. How connected is a single node? And ones that are more highly connected, okay, we'll pull those out and we'll stick them into the longitudinal model that includes all the other variables that are important to us. Does that make sense? Yeah.
Question about your correlational model.
Yeah.
And that is, when you built that model, do you think that you could also utilize that model to figure out how a perturbation for any particular part of the system would impact other parts and simulate that out? The reason I ask is you mentioned that it may not be enough to give an intervention, like to say put a certain microbial agent back into a system. You can't really predict what the impact will be.
Yeah, so the networks, we used an undirected network. So you'll notice the networks don't have any arrows on it. But you can actually do directed networks. Again, these are tools that have been around for a while. If you have, the tricky part is you need longitudinal data, which we had, but you also have to have, this is just one of those irritating parts of using some tool, but, or technique, is that you have to have information on all of the same taxa. Or, every single variable has to be measured at every time point. So if you have, for example, at time point 1, you have 53 taxa. At time point 2, you have 72 taxa. At time point 3, you have 19. You're only going to be able to use 19 if they all overlap, right? So it frustratingly reduces it. Now, I'm not sure if there are, I'm not an expert in network analysis. It may be possible to explode that and be able to use what is essentially missing data at each of the time points to generate those directed networks. But that would be one way. If you had an intervention at one time point and you could look over time, you would see how those relationships change and you could know which direction they change in and the magnitude of the correlation or covariance.
or in your case, there's case versus control. But are there already fixed ones?
Oh.
People have
Yeah, so I mentioned that people have done studies trying to define enterotypes. So these are like gut communities that seem to be characteristic of certain geographic areas or cultures or things like that. I'm not a big subscriber to that. Because if you asked me whether there were enterotypes in Vancouver, can you imagine how many enterotypes there could be here? Based on the dietary diversity, the age distribution, the close contact with dogs, you know, like, I mean, I think it would be really hard. And the other thing is that these are very dynamic. Okay, I will say, however, that there are major members of the microbiota, and they do have certain functions, right? You have Ruminococcus is a good degrader of fiber. So if you have somebody who has a very high fiber diet, then you're probably going to see Ruminococcus or Ruminococcus-like organisms there. So there may be common organisms that are characteristic of certain very common exposures, right? And diet is the big driver in every case. So you may find common characteristics among people just because fiber is a big part of a lot of people's diet.
I actually meant common networks.
Oh.
So you do network analysis with certain bacteria related to, is there a fixed?
Okay, oh, I see. So the relationships.
Always
Yeah. So as far as I know, there's only been about 3 studies that have used networks in microbiota analyses. So it's still pretty new, which is surprising to me because abstractly you'd think that the microbiota should probably be functioning network-like. And so this might be a good tool to use. But no, yeah. Yeah, I think people mostly, it's changing now, but mostly there's been a lot of characterization of who's there and in what abundance under very different conditions. But there hasn't been a lot of how do those members relate to each other. Yeah. Yeah. I'm sorry? Other than glutamate? No, not yet. Yeah, no, so we're going to do that. So the trial that I'm working with is this huge trial, 5,200 households. And we're going to try to do metagenomics, but you know, as everybody in the room who knows about microbiome research, you know that the composition is not where it's at. And measuring function is always a challenge. So we're doing composition, we're doing metagenomics, but we're going to do metabolomics and try with both of them. First, know which organisms are active in the gut. So sometimes up to 50% of the DNA signatures you get from the metagenomics are from dead, damaged, or transient organisms. They're not real members. So this is a big weakness, not just having the stool, but the fact that what's in the stool isn't even a good, accurate representation of which organisms are active in there to begin with. So you need to pair good compositional information with the functional information, and that's happening more and more, especially in the experimental models. Yeah. Okay. Shared pathways for. Yes. Yeah. Yeah, so you're thinking maybe of mapping the correlation structure with the underlying pathways. So you know which organisms are related in abundance. You might be able to see, well, correlated or inversely correlated. You would see the underlying pathways related to that. Unfortunately, a lot of bacteria have similar pathways, but you have some like Acidaminococcus and Ruminococcus that have, you know, they're really the heavy hitters for that functional purpose in the gut. So you may be able to do that for certain organisms that have distinct functional characteristics. Yeah. Yeah, so I think, like I said before, knowing, I think for the kids especially, the EED, the environmental enteric dysfunction, that's really characteristic of the small intestine, right? The community is important there. I don't think we know enough because we simply can't get biopsies of enough people in the different regions of the gut to know whether the populations are more discrete or they're continuous. In some ways they're continuous, but there's some organisms that are clearly colonizers of specific areas because they have a specific function there. Yeah, so we don't know. And I told the students earlier that it would be ideal if people could swallow tiny robots and that they could pass through the intestinal tract and collect samples, you know, maybe at random, but all the way down the gut and come out the other end so that we would have an accurate picture of both the tissue and the organisms that are associated with it. So, and I think everybody, well, I agreed, maybe some other people agreed, that people would be happy to let colonoscopy go forever if they could swallow tiny seed-sized robots. So anybody has an idea for how to develop that, I would be very happy to be involved in that, especially before I turn 50 and I have to undergo my own colonoscopy. Any other questions? Yeah?
You mentioned that you don't trust the fecal swabs as an accurate representation of the gut microbiome.
Yeah.
What evidence do you have?
There have been a few studies that have looked at biopsy samples along the intestinal tract in small numbers of people, and the stool does a good job, but not a great job at capturing the whole, the level of diversity that exists along the entire intestinal tract. It's just simply not possible to.
Is it more of like the later fact that
Yeah.
with the fecal swab?
Yeah, yeah. And you're not getting, I mean, you're getting some organisms that are totally tightly associated with the mucosa, but you're not getting very many of them. You're only getting the stuff that's being sloughed off and that's being passed in the stool. So if people have looked at trying to partition the stool and try to get mucosal or cell-associated microbes, it's very difficult to do on those samples. The other thing is trying to do a washout, right? So often you have a bowel preparation before colonoscopy where kind of large volumes of fluid come out. And if you could take samples of that fecal water or the fluid that's passed kind of toward the end of that clean out, you might be able to get more cell-associated bacteria. But that's also not entirely, that's somewhat invasive also, so yeah. Yeah.
How much work are food companies doing in this area? I know on TV we're challenged on the
Oh,
challenge.
yeah. I think they're interested. So the issue is that functional foods and active, you know, microbial products, there's pretty a narrow list of ones that have been approved for use in food products. And the microbes are pretty wimpy, the probiotics. That would involve a lot of testing. If you went back and you took, you know, for example, as far as I know, there's no ruminococcus probiotic. But if you found a candidate, you would have to do all the safety testing in order for something like that to be able to go in a food product and be distributed widely to everybody who's healthy. So that may be possible, but it would be a huge amount of work. And so they kind of continue to work on the same small set of bacteria that have been approved for use in food. So it's a pretty small group. But, you know, probiotics, I've always been very suspicious because if you think about it, you have 10 to the 12th organisms in your gut. And here you have a wimpy lactobacillus. And it arrives in the gut as it makes it, hopefully through the stomach. And it arrives in the gut and says, oh my gosh, who are all of you? Like you can't really compete. Well, so if you're taking it every day, maybe it has an effect. But as soon as you stop taking it, it can't really, they're not very good at gaining a foothold in competition with 10 to the 12 other organisms per gram of stool, right? So I'm a little suspicious of that. Now, if you did a colonoscopy, like if you did a bowel cleansing completely, please do not misunderstand me. I am not suggesting that anybody go through this. Let me just be very clear. I'm not a physician, but just thinking colonizationally, if you really reduced the numbers of organisms, and then you took a functional organism like a probiotic or something like that and tried to call it, maybe it would have a better chance, you know, because the numbers of organisms would be lower. something like you would have to really perturb the microbiota first, like take heavy doses of antibiotics to clear it out. Then you may have some organisms that can permanently alter the composition, but just providing a probiotic in food, while you're taking it, it might actually work, but then it'll gradually, the effect will disappear. That's just my hunch, because you can't compete, right? And so, yeah, the marketing is a little dubious. So I'm happy I'll stay if people have questions. I know he does. Great, thank you.
Thank you so much, Dr. Manges, and thank you everyone for coming tonight. I'd like to invite you all to join us out in the atrium/lunchroom for some pizza and beer. And next VanBug will be November 3rd, I believe.
Yeah, no, actually, the deck was done on all of that.
Okay.